All right, this is the biodiversity inquiry, which is based on the phenomena that arthropod biodiversity is declining both locally in our areas as well as globally in other places around the world. So when we're focusing on arthropods, think about insects, spiders, crustaceans, small crabs, certain types of worms. It's a group of animals that's pretty large that do not have a backbone. So the focus here is to be able to do at least 2 collections from 2 different places based on a variable that you or your teacher may be choosing or give you a choice from a list. It could be things like 2 locations that are different in terms of light, amount of sunlight, light versus shade, amount of moisture. What we focused on for this particular example that we're doing would be the effect of human impact on the environment. So we went to 2 locations in Eastern Oregon on the campus of Eastern Oregon University. And we looked at one location that was literally next to a very busy parking lot. So you have lots of foot traffic, lots of automobile traffic, exhaust. About 200 feet away, closer to a building that was not next to a parking lot and definitely not a busy parking lot, we did a similar sample of leaf litter. And so we have 2 samples to look at and try to analyze and determine based on the arthropods that we find in those samples what the biodiversity is for each of those locations. And that's what you're going to be able to do once you start seeing what was found at those 2 locations. So just to give you a quick idea of how the sampling occurred, the leaf litter samples, basically that includes leaf, dirt, pine needles, wherever you happen to be. We have a lot of pine needles here in Eastern Oregon. But each sample will be collected in a pillowcase that's breathable. So if you, for some reason, were collecting and you weren't able to identify the arthropods on the same day, it would be okay because the material that most pillowcases are made from is pretty breathable and air is able to get in and out. But we take the pillowcase for whatever site we're at and we put it on top of a 5-gallon bucket like this one. And there are different types of sifters. Most people just make their own homemade. This is just a box from a grocery store with some netting at the bottom. And you put the sifter on top of the bucket. And then once you find your area that has leaf litter, you will reach in, grab the leaf litter, put it into the bucket. I tried to keep everything controlled as much as possible. So I put in 3 clumps or handfuls of leaf litter for each of the sites. And then you'll start to shake it back and forth. And a lot of the bigger pieces will get netted out basically. And then hopefully some of those arthropods are going to go into your pillowcase. And the goal is to try to keep everything as controlled as possible. So think about some controls that you might want to be aware of. How long were you shaking each time you had leaf litter in there? And was it the same for each of the different locations that were tested and sampled? Think about making sure that you do the sampling on the same day, because from day to day you might have different temperatures, different amounts of moisture, different wind speed, things like that. So it's just something to keep in mind. And then once you're finished with the actual collection, you take out the pillowcase and just do a quick little knot at the top. And then once you've got your knot at the top, that should keep everything inside. You're able to take it back to wherever they're gonna, most of the time it's a classroom. Some teachers may have you actually do the recording and identification out on the field. It does help to bring some type of a clipboard, notepad, so you can make observations about different abiotic factors that were in play when you were doing the sampling. You may need a tape measure if you're trying to measure between 2 distances. The other kind of key thing also is to wear gloves that are pretty heavy duty, because there are stickers. We had lots of pine needles that would poke your finger and hurt pretty bad. And then you also want to make sure that you're labeling with some type of a sticker or label where that sample came from, because you're going to need to look at and compare at least 2 different locations. Sometimes, depending on what your teacher asked you to do, it may be 3.